Hey, Honors Bio people. Here's your quick flipped video on basically on the movement of materials, or the process at which the materials that you need to get in and out of your cells get in and out of your cells through that cell membrane that we learned about yesterday in class. A couple things first. There's a couple terms that we wanted to make sure you guys understand the difference of. One of them is called selective permeability, and the other is semi-permeable. They kind of go together, but the idea is that if something is selective, has selective permeability, that it only allows real specific things through that membrane. And semi-permeable is more or less the same thing, but again, it's the term where it works like almost like a filter, or like the screen on a window. It'll let certain things through, like the screen lets air, the wind, maybe rain through the screen, but it does not let insects or birds or an elephant through that screen. It's semi-permeable, and that is exactly what your cell membrane is semi-permeable. It has selective permeability, same thing. So what I've created here for us is a flow chart just to show you the basic kinds of transport. We'll start with one that is, well, there's two versions. There is what's called passive transport, which follows the concentration gradient, and then there's active transport, which goes against the concentration gradient. This one requires energy. Passive is free, doesn't require it. Now, passive is broken down into two parts. So we have simple diffusion, right? Diffusion is when you're talking about real small molecules, things like oxygen or carbon dioxide. They're nonpolar. They can go right through the membrane, no problem. Now, we also know that if you're talking about water, then it's a specific type of diffusion that is called osmosis. All right? The other version of passive is also called diffusion, but it is called facilitated diffusion. Now, if you know what the word facilitated means it's basically helped. It needs a helper. So there has to be a protein channel. These are things that are larger, and kind of the classic example is something like glucose. It's too big to fit through the membrane, so it has to be helped, but it still travels from high to low concentration. On the other side of the spectrum, with active transport, this is where we would have specific protein channels, and this is where it's going against the concentration gradients. So we have proteins, and we require ATP, or energy. These are things, kind of the classic is this sodium-potassium pump, which we'll mention briefly. And then the other version is on a large scale, and it's something called endo or exocytosis. So endo or exocytosis. And that's basically when you have something very large that needs to get in, endo, or out, exo, of the cell. The membrane will actually pinch itself off and create a little vesicle of it in and of itself. So those right there are the different types of transport that we're going to take a quick look at. Diffusion is real simple. It's simply the movement of something from high to low concentration. So I often think of it like it is a hill, all right? This is your high concentration, and this is your low concentration. Whatever that particle is going to start at high, and it's going to work its way down to low concentration. It's free. It's rolling down the hill. It's following the concentration gradient. Again, this is things like oxygen and CO2 can do this. Here's a little better picture of it showing you, it's highly concentrated over here. It's low concentrated over here, and it will spread out until it is even. That particle, that little green dot, is the solute. It's the dissolved substance. It could be a bunch of different things, but it's whatever is dissolved in that solution. So that's simple diffusion. Osmosis, we already mentioned, is diffusion, but it's diffusion, and the key here is that it is water. It's only water. Now, this is where that semi-permeable membrane comes in. This membrane right here is semi-permeable. That means that certain things will pass through it. In this situation, water will pass through it, but the little solutes will not. So water is going to move back and forth trying to balance out these things. And in the process, it creates different types of solutions. Something called hypotonic or hypertonic, or ideally, what's an isotonic or equal solution. So let me show you a little bit more. This is something I saw. It actually made pretty good sense. They call it the U-tube experiment because it's a tube that has a sense. So the membrane right here is semi-permeable. The solute, let's say that the dots there are sugar. And the water would start out like this. Now you have an equal level of water. You have a semi-permeable membrane. The sugar cannot cross that membrane, but the water can. And what will actually happen is that because there's a high solute of water on one side, right, on this side over here, it's a bit more pure water than it is on the other side. What the water will do is it will flow through that membrane and try to dilute out that. So the water will sort of magically rise on one side and lower on the other, with the idea that it's going to dilute out that solute so that it is equal on either side. So the water would move from one side to the other until it is equal in concentration. It's kind of a neat little thing to show osmosis and how it works. All key is the semi-permeable. All right, so what you get as a result of that is you get cells, and this picture should look familiar. We saw this the other day. But what you get is cells that will either shed water or gain water to try to make the solutions equal. So a hypertonic solution is one that has a high amount of solute, so it has lots of dissolved particles. Hypotonic has very little, and isotonic is equal inside and outside the cell. Now your cells are they're going to move water back and forth. And what you would see, and this is a scan out of one of our books, but what you see here on the animal cells on the top is that if this cell, say we drew a beaker here, and this cell is in a hypertonic solution. In other words, there is tons, we'll do it in black, tons of particles outside this cell. Just tons of it outside and very little inside. What happens is that is a hypertonic. There's lots of solute. So the water will be flushed out and the cell will shrivel up. And if it shrivels up enough, it will cease functioning. So a hypertonic solution will give you shriveled up cells. It even happens inside of the plant cells. An isotonic solution means that if we put it in a beaker, it would have equal amounts of solute outside and inside. Doesn't mean the water doesn't move, but what it does mean is that for every water molecule coming in, there's a water molecule moving out, and it's at a balance. Iso is equal, it's at a balance. The other version is the hypotonic solution. Now this is what happened to those little paramecium in the video I showed you guys. There's your beaker. What happened is there was a high concentration of solute inside and not nearly as much outside, so the water, because the solute cannot pass through the membrane, it's semi-permeable, the water will jump in and it will swell up to the point of actually bursting or lysing. Remember, lyse means to break. It'll lyse that cell and actually cause it to explode or blow up, much like that paramecium did in the video you guys saw in class. Right? How does this work for us, or what does it do to us? If you take a look at the pictures I pulled here, these are what happens. These are actual red blood cell pictures underneath the microscope. You can see here in an isotonic solution, equal amounts go in and out. The size stays constant. Cell is happy. Hypertonic solution, which if you remember, if it's in a hypertonic solution, that means there's a lot of solute on the outside. So this would be a situation where your body is severely dehydrated. Your cells are going to shrivel up or possibly even die. That's dehydration at its maximum. In the hypotonic solution, there's a lot of concentration inside, nothing outside or very little, so water goes in and causes the cell to swell up and even burst. This actually could be if you are overhydrated. Okay? Obviously, this is the one we focus on more often than not, but overhydration can happen. Some examples in your body, other than that bloodstream, diffusion through a semi-permeable membrane. That's what happens in digestion, the way you absorb the nutrients in your body. The food you ate, there's a higher concentration of nutrients in your esophagus, and it passes through the wall and out into the bloodstream and to get brought around the body. Happens in respiration. When you breathe in, you have a higher concentration of oxygen in your lungs, so it will push from high to low that oxygen right here out to your bloodstream to oxygenate your blood. Same idea with the waste coming out. They're all just versions of simple transport, okay? We talked about passive. Key is right here. Without the use of energy, it is concentration driven. Remember our hill, high to low concentration. That's passive. Diffusion, that's the lipid bilayer right here. You notice there's no proteins. We don't need them for this. Things like oxygen, carbon dioxide, small lipids, alcohol, things that are lipid soluble will pass right through from high to low concentration. Free. Facilitated, it's still free, but there's too big to fit through, so they must go through one of these specialized channels. They're still moving from high to low, but these are things that are larger and or polar molecules like ions, proteins, glucose, salts, water-soluble vitamins. Water actually goes through a special type of protein channel called aquapore, or aquaporin. It's a special protein, so water can jump across that membrane. Facilitated diffusion, examples of that. How it works, specialized channels. They have to fit. If they don't fit in the entrance, they can't get through. Some of them are gated like this, where they have to bond inside of it to open it up. Still, high to low concentration. It is free. So if you look on the sheet here, right here, that would be an example of simple diffusion, high to low. This is a channel protein, so that's facilitated diffusion, where it's just sliding through. That's how water would get through. This is a little bit larger even, still called facilitated diffusion because it's going from high to low concentration, and these are even larger, so they have to go through a larger channel. Like we said, was called active transport. This is materials going against the concentration gradient, so it requires the use of energy. So in other words, in this case, right, low and high concentration, we're actually pushing against the concentration gradient by coming in. To do that, it requires energy to push it through. The classic example is the sodium-potassium pump. Sodium needs to go out against the gradient, so sodium loads up inside. ATP then pushes it through. As ATP pushes one through, it allows potassium to come in the other way. So it's the use of that energy that causes that to happen. This is against the concentration gradient. ATP, we've already mentioned this a few times, but ATP, adenosine triphosphate, that is the energy molecule. Basically what happens is the energy is released when you remove a phosphate, then the energy comes out. This can be reloaded and reused again, and your body will continuously use this to cycle things through your membrane. It's called ATP. We'll get more into that in the future here in the class. The last one I mentioned, I'll just show you again one more time. Exo and endocytosis. Exo, think of exit, so it's moving large materials out of the cell. One classic example is when those lysosomes, right, those organelles that clean up the inside of your cells, they have these vacuoles full, or vesicles or vacuoles full of sort of waste products, and they need to remove it from the cell. So they will essentially slide over to the membrane, bond to the membrane, and then that becomes the new membrane as they release that waste product. That bonding requires energy. But that's exocytosis, moving large materials out. This is endocytosis, which is the exact opposite. You're moving a large material in. Something very large needs to get in right here. So what happens is the membrane will start to pinch off and envelop it, and then it creates this vacuole inside of the cell which contains that large material. So just giving you that quick rundown of cell transport, active, passive, and then the different versions of. Hopefully that helps. Thanks.